Yesterday, Dr. Meyer laid out the kind of all the definition of evolution and intelligent design. And today, he's going to have further evidences that support that. Hopefully, that we absorb everything we can, and be able to have a very intelligent conversation with our friends, so we can introduce Jesus through all this. Amen. All right. So without further ado, we have Dr. Stephen Meyer.
We'll have a little science seminar this morning. This will be, of the three things I do, we'll have the most science in it, but tomorrow we'll have a lot on cosmology and the origin of the universe as part of a talk on Romans 1, so that'll be something new altogether. I promised last night, I was going to give I've written 3 books. The first was called Signature in the Cell, which is about the evidence for design in the digital code that's stored in the DNA molecule, and the puzzle that information has posed to scientists trying to explain the origin of the very first life. The long-standing question is, how do you get from the chemicals in a supposed prebiotic soup to the first living cell? And that's what my first book was about. The second book was about the origin of the first animals. And this is a question that was addressed by Darwin's theory of evolution, but has been left unanswered. Darwin himself knew it was a mystery, and we'll be talking about that this morning. And so, what I decided to do is to change and talk about this second book today, because there's a really interesting Chinese angle on this story, and Pastor thought that people would find this particularly interesting, and it's fascinating in its own right. And in tomorrow's talk, I'll talk a little bit about the case for intelligent design from the digital code, as I did last night as well. So this will give us just a little bit more exposure to some of the things that I've written about. So there was a very exciting, to start today, there was a very extraordinary, let's see, maybe I'm too far away to make it click, extraordinary news story in 1995, made the cover of Time magazine, and it concerned an amazing fossil find that had been made in southern China in a place called Chengjiang in a formation called the Maotian Shale. And this was, as the Time magazine article says, it was a frenzy, a biological frenzy that was documented in the fossil record of new forms of animal life at the very, the earliest part of Earth's history where animals came on the scene. It's an event known to paleontologists. It's called the Cambrian explosion. The Cambrian explosion. And we're going to learn about the Cambrian explosion today, and it's mysterious for a number of reasons from a Darwinian point of view. So first of all, let's just define it. What is the Cambrian explosion? Well, the Cambrian explosion is the abrupt or geologically sudden appearance of most of the major animal groups and their corresponding body plans that have ever existed on Earth. So we got to unpack that a little bit, but you can see some pictures there of different forms of animal life, arthropods, chordates. Chordates are animals like us that have a spinal column or a notochord. Then you can see many other different types of worms, brachiopods, some jellyfish-like things. And these represent what are called the phyla. So if you had some biology back in the day, you might remember a little acronym I was taught: Kings play sand on fine green, Kings play chess on fine green sand. Okay, did I get that right? Ideas. You have the species, the genus, the family, the order, the class, the phylum. As you go up the hierarchy, the categories of biological similarity get bigger and bigger. And the highest category, the highest classification of animal life is called the phyla, and the phyla correspond to unique body plans, where a body plan is a unique combination of body parts and tissues. So let's take the arthropods there do you see the iconic image of on your right, the trilobite over there? I loved those when I was a kid. I loved dinosaurs, and I loved trilobites, and the trilobites have these three body lobes, and they have a hard exoskeleton. All arthropods have a hard exoskeleton, and we have arthropods today, like crabs, okay? So that if you have hard exoskeleton and all the soft parts are on the inside, that's a certain kind of body logic, okay? If you put the skeleton on the outside, you got to arrange things differently on the inside. If you put the skeleton on the inside, as you do with chordates, then that's a different kind of body design. And so what biologists have noticed from the time of John Ray and back in the 17th century, probably going all the way back to Aristotle, is that there are different types of organisms that have a different kind of logic to their arrangement of body parts that makes them work as a whole organism. And so in the Cambrian explosion, over 2/3 of all the body plans that have ever existed on the planet first came into existence in that burst of biological creativity. And what happened in southern China in the 1980s and 90s, finally making it into the mainstream media in the United States, was that was the discovery of one of the greatest Cambrian fossil finds in the world. Previously, the Cambrian fossils had been known by Darwin and some of his contemporaries in a formation in Wales. And then there were other formations. A great formation was found in British Columbia called the Burgess Shale. I've had the opportunity to hike up to the top of it to see the fossils. It's an awesome thing if you get a chance. And we've sent a group of our scientists to China, to Chengjiang, to see the fossils there. But this was such a big deal. In fact, in that Time magazine article, the abruptness of the appearance of all these new forms of life was so dramatic that one of the Chinese paleontologists said, how fast does this have to happen before we stop calling it evolution? Boom, there they are in the rock record. Thousands of feet of pre-Cambrian rock, no animals, or very simple things like sponges that are an animal. But then suddenly you get these complex animals arising very abruptly. That's the exciting and extraordinary thing. Now, we can get to kind of a picture of why this is so dramatic. Here's a picture that helps explain this. You have the standard geological time scale in millions of years on, let's see, that would begin on your left, and then you've got the sedimentary rock column on the right, and this is just, idealized a little bit, but then you have all these new organisms arising very abruptly with no apparent ancestral forms in the lower pre-Cambrian strata. And that's the mystery, because if you remember what we were talking about last night about Darwin's theory, you remember that he represented the history of life as a great branching tree. And so if on the expectations or predictions of his theoretical framework, you would expect to find the first animals anticipated by slightly simpler versions of those same animals in the lower pre-Cambrian rocks. So if you get the first animals in the Cambrian, and they're represented by the gold dots, you'd expect to find a lot of other similar things, maybe just a little bit simpler, a little bit simpler, a little bit simpler as you go down in the Precambrian rocks represented by the blue dots. But the problem is no blue dots. There are no Precambrian animals. In my book, Darwin's Doubt, I go through this with real technical precision. And depending on how you count, there are roughly about 36 phyla in the history of the planet. I think the count was about 27 arise in the Cambrian. There are 3 that arise in the Precambrian, but they're very different than the things that arise in the Cambrian, and one of them is a sponge, very simple. So this is a really dramatic appearance of major new biological form. Okay, so this is the data, if you will, the abrupt appearance, and then this is the data compared to the theory, and you can see there's a disparity. And this bothered Darwin even in his own time. He said, as to the question why we do not find rich fossiliferous deposits belonging to these assumed earliest periods prior to the Cambrian system, I can give no satisfactory answer. And in my book, I called this Darwin's Doubt. He did not doubt the adequacy of his theory, but he doubted its ability to explain this data. This was an unresolved mystery from his standpoint. And it turns out that Darwin was a lot more forthcoming in acknowledging weaknesses in his theory than his modern proponents. I sometimes call them Darwin's public defenders. I had a chance to testify before the Texas State Board of Education a few years back, and it was about public school curriculum. And we were arguing, don't worry about our theory of intelligent design. You don't need to get that into the schools if that's going to cause everyone to double over with anxiety and initiate lawsuits and that. Let's just start by presenting not only the evidence for Darwin's theory, but also the scientific evidence challenging his theory, as you can now read about it in the peer-reviewed scientific literature. How about that? That seems a reasonable thing. That's just a matter of scientific literacy. Scientists are talking about problems with Darwinian evolution in technical peer-reviewed articles. Can the students in our public schools not know about that at least? That was our proposal. In response, the National Center for Science Education, which has a little shop up in Berkeley, California, sent Dr. Eugenie Scott, and she was quoted in the Dallas Morning News before this hearing as saying, there are no weaknesses in the theory of evolution. You can't teach the strengths and weaknesses of competing scientific theories, at least not when we're talking about evolution, because there are no weaknesses. Now, I hope by the end of the lecture you'll see that that's a somewhat laughable statement. Sorry to be feisty. Similarly, Richard Dawkins, another great public defender of Darwin's theory, says it's absolutely safe to say that if you meet somebody who claims not to believe in evolution, or evolution with a British pronunciation, that person is either ignorant, stupid, or insane, or wicked, but I'd rather not consider that. I thought that was so sporting of him not to consider the hypothesis of wickedness among those of us who raise questions about Darwin's theory, because after all, Darwin himself raised some questions about Darwin's theory. But his public defenders in present do not do that as a rule. Actually, there are groups of people who present evolution for public consumption that don't do that. Technical biologists working in evolutionary biology are acknowledging lots of difficulties with the theory, and we'll learn about some of those today. So Darwin acknowledged this problem, but he had an idea about how it might be solved. The problem of the absence of ancestral precursors to the Cambrian animals. I call that the mystery of the missing fossils. Where are those ancestral forms? He said the case at present must remain inexplicable and may be truly urged as a valid argument against the view here entertained. But he went on to say about this mystery that there might be a way of solving the problem. He said, I look at the natural geological record, he meant the fossil record, where we have the sedimentary layers of rock representing different ages on planet Earth and different fossils in those different layers of rock. He said, I look at the natural geological record as a history of the world, but one that's imperfectly kept. On this view, the difficulties above discussed, meaning the Cambrian explosion, are greatly diminished. Here was his idea. He had this charming illustration likening the layers of rock to chapters in a book, where some of the chapters, for whatever reason, had kind of been torn out. And so we only had a record of some part of the history of the Earth, but not all, because for some reason or other, the fossils in some periods of time were just not preserved. Okay? So they were either preserved or else we hadn't found them yet. Okay? And this became known as something called the artifact hypothesis. And the idea of the artifact hypothesis was that Darwin's tree is correct, but if it looks like a lawn in some places where there's no branches beneath, then that's because we either haven't found the fossils or we haven't, or they weren't preserved. The missing fossils are an artifact of incomplete sampling of the fossil record. We haven't looked hard enough, or they're an artifact of incomplete preservation. Okay, so that was the idea. It's a reasonable, what's called auxiliary hypothesis, a hypothesis that is formulated to support a difficulty in an original hypothesis. So that has seemed kind of plausible for a while, but by and by, the Cambrian explosion became more explosive. Well, at least from our point of view. It was always equally explosive in the actual history of life, but we realized that it was more explosive than we even knew in Darwin's time. And so in 1909, there was an extraordinary find in Canada called the Burgess Shale. And there were this is a picture of some of the trilobites that were discovered there. And there was a whole array of Cambrian animals, including some animals that we had not been preserved in the formations in Wales, about which Darwin knew. And so there were trilobites and there were brachiopods. Oh, actually, the point of these first two slides is to say those are the two animals that Darwin knew about from Wales. But when you got to the Burgess Shale in Canada, all kinds of new critters showed up in the Cambrian strata. And so what we realized was that the explosion had been even more explosive than we thought. And this is another kind of arthropod called a marilla. It's a delicate little lace crab, and it's beautifully preserved in the rocks. Here's an artist's rendition of this little creature. This is a guy called Waptia. It's a shrimp-like thing with a hard head shield with exoskeleton. So lots of different animals that had not been known before. And this is a crustacean in the subphylum of the phylum Arthropoda. So lots of cool stuff. Now, so that made the mystery a little deeper. And when these Cambrian fossils were found in the Burgess Shale, again, no ancestral precursors. So it's getting a little suspicious here. Everywhere we look, you get more and more Cambrian animals, but still no transitional intermediates below. So it doesn't seem to be a function of just one locality. It seems to be the same pattern all over the world. Now, this little dude, we've got some animations. This is a guy called Opabinia. He was discovered at the Burgess Shale. He's got 5 eyes. He's not the intelligence network of Australia, New Zealand, Canada, Britain, and US. No, he's got 5 literal eyes. And he's got a long proboscis with a pincher on the end and just exotic creatures. Stephen Jay Gould wrote a whole book about this called Wonderful Life. These weren't known from Wales, but the Cambrian explosion turned out was much more explosive than we thought. So I've got a little animation reel of a whole bunch of these guys that were discovered, but we'll move on, you get the point. Now, next big fossil find. This is the find that was discussed in the Time magazine article. This is from Changjiang in China. Pastor Keith, did you say last night that you guys had been to that part of China at some point in the southern? Yeah, it's fascinating. We sent a group of scientists there. There's a scientist at the University of San Francisco now named Paul Chien, and he's connected to us at our institute, Discovery Institute. When he read about this in Time magazine, he reached out and connected with the leading Chinese paleontologist, a paleontologist named J.Y. Chen. And Paul went over there and was able to go to the site, see the fossils. He was an expert on embryos, and they had found some small, tiny sponge embryo fossils. And so he did a lot of analysis on all this, and we eventually sent a group of scientists to a conference there in 2001. In any case, the Changjiang biota are now world famous. And this is Professor J.Y. Chen from China. And there was an article actually, well, let me tell you about again, the big story here is, first of all, the beauty and precision of the preservation. One of the first of the Chinese paleontologists in his writing said it was almost as if you could see the animals preserved perfectly on the wet surface of the mudstone. It was like they were almost alive, and yet with our standard dating methods, they would be about 530 million years old. And so this was another arthropod that they discovered. It was the top of the food chain predator in the Cambrian seas. These were all, by the way, marine invertebrates. They were all animals that lived in the water. And this was called Anomalocaris. He had little pinchers. He was about 1 meter long, a large arthropod with pinchers that chased down other critters, I guess. This is a hyolith. Had a conical shell with an attached lid with a little animal that lived inside. This was a filter-feeding worm that had a muscly foot that allowed it to attach to the bottom of the seafloor, and there was a worm that lived inside the tube called a Foronida. This is another type of worm with a completely different body plan called an annelid worm. And this was a strange creature. They didn't know what it was exactly, so they named it a problematica. Now, this is how science is done, folks. You know, if you don't know something, give it a fancy sounding name, and then it's all taken care of, okay? Phylum Problematica. They think it was probably an animal that looked a bit like a plant, and then it had these petal-like bracts and a feeding bowl, and then it, again, attached to the seafloor. I love these guys because, well, for a couple of reasons. We have them in Puget Sound in the Seattle area. The phylum is Ctenophore. They're like, kind of like jellyfish, and they have a translucent body, a jelly body with biradial symmetry. They've got these ciliary comb plates, and the one on the left, on the left is is a modern version of the same creature. They're comb jellies. The one on the left is a modern version. You can see the artist's rendition of the fossil showing that they haven't changed in 530 million years. So this is another part of the mystery. Stephen Jay Gould called it stasis. You get these new body plans emerging abruptly, and then they persist in the fossil record over long periods of time without changing at all. Where is the evolution, folks? Well, with many body plans, zilch in 500 million years. And this is an example, a dramatic example of stasis. And even since the 1995 article in Time magazine, the very first primitive fishes have been discovered. So we actually have examples of the chordate body plan very, very early on in the animal fossil record. So you get this dramatic burst of biological innovation, what the biologists call morphological innovation, where morphology is the study of form. So it's really dramatic. So from Darwin's time to the Burgess Shale discovery to the Chengjiang discovery, we have learned in progressive episodes that the Cambrian explosion was ever more dramatic and explosive than we realized. So that's a big part. That's first big lesson of this Chengjiang find. But the other thing, the beautiful preservation, the abrupt appearance, the disparity of form that was unknown prior to this, but also there was another aspect of it, and it bore directly on the question of the artifact hypothesis. Oh, by the way, this is a lovely quote from Dawkins where he's being a bit more honest. He says, "It's as though they, the animal phyla, were just planted there without any evolutionary history." Thank you, Richard. But are you being wicked by saying that? I don't know. That wouldn't be nice to say that. Okay, so remember the artifact hypothesis. It's the claim that the ancestors were seldom fossilized because they lacked hard parts. Okay, the first version of the artifact hypothesis by the 1980s and 90s was wearing a little thin. We'd have, by that time, well over 120 years to look for new fossils in the Cambrian, and the globe was being scoured by paleontologists, and you just don't find the ancestors to the animal forms that arise in the Cambrian in the pre-Cambrian strata. They're just not there. And that's still the case in 2023. So that version, but the version that seemed to get some traction was the idea, well, maybe there was something different about what's called the depositional environments, the way the sediments formed in the pre-Cambrian that prevented the ancestors of the Cambrian animals from being preserved. Or maybe it was because those ancestors lacked the hard parts that came later in the Cambrian period. Well, that idea was kind of put to rest in the Chengjiang, with the Chengjiang find, because there was a layer underneath the Chengjiang in the very late pre-Cambrian called the Dushantu Shale. And in that shale, there were little tiny microscopic fossils discovered. Now, there is currently now some debate about this. Initially, the analysis that our own colleague J.Y. Chen did identified these as sponge embryos. There's now some debate about that, but what's not in debate is that these were soft-bodied embryos of some kind, microscopic. So that raised a big question, because in the layer of rock just below the layer that documents the explosion, you're preserving small, soft, microscopically small, soft tissue. And if you can preserve that, then why aren't you preserving the hard parts of a trilobite, or the skeleton of a fish, or any of these other larger animals that came online in the Cambrian? What's the problem? If you can preserve small, soft, microscopic tissue, why aren't you preserving the exoskeleton of a Cambrian trilobite? They're called carapaces, and they litter the fossil record. They're easily preserved. And so as a consequence of this discovery and others, showing that soft tissue was readily preserved in the Cambrian and in the Precambrian, you have to ask, this has posed a huge challenge to this artifact hypothesis. And in 2013, the same year my book came out called Darwin's Doubt, a book by two leading American paleontologists, James Valentine and Doug Irwin, said, look, the artifact hypothesis is dead. Whatever else we make of it, we have to treat the Cambrian explosion as a real event. It's not an illusion of sudden appearance. Things really did appear that abruptly. We've got to explain it some other way. We can't cite missing fossils because both versions of the artifact hypothesis have gone down. Okay, so that's just to say that the mystery of the missing fossils from a Darwinian point of view has gotten more mysterious, much more mysterious. It's unsolved from a Darwinian point of view. Also, by the way, note that this evidence taken at face value does not support the tree of life picture, the history of life that we talked about last night. That was my second definition of evolution. Remember last night we said the first definition is just change over time. Second is the idea of continuous change over time, so that the history of life is best depicted as a great branching tree. And then the third definition was the idea of an undirected, unguided process that produces that change over time. Well, even evolution number 2 is a bit challenged, significantly challenged by these findings, because the picture of the history of life you get from the fossil record is one of discontinuous change, not continuous change, but discontinuous change. It's abrupt appearance, not gradual morphing of one thing into another. Interesting in its own right. Okay, now, the second, and in my book I argue more significant mystery, is actually the mystery of how you build these animals. Or how, let's be more precise, not anthropomorphize, how the evolutionary process would have generated all this new biological form, especially when we consider how abruptly it did so. The Cambrian explosion is typically dated about 10 million years, as a 10 million year event from beginning to end. Now, that sounds like a lot of time, but that is geologically extremely abrupt. If you think of the history of the planet's 4.6 billion years old by standard dating methods. The first life comes at about 3.85 billion years. The Cambrian animals arise about 530 million years, and they do so in a very narrow window of time. It's about like 4 inches on a football field is the abruptness of the event, okay? Now, that still sounds like a lot of time, but it turns out that it's not just geologically abrupt, it's also biologically abrupt. There is a branch of evolutionary theory known as population genetics that allows biologists to calculate how much change they would expect to occur in a given amount of time if they know certain parameters, like the mutation rate or the population size, and so forth. And it turns out that 10 million years is not nearly enough time to generate the massive amount of new biological form that arises. It's the evolutionary process simply doesn't work that fast. Even if you assume that Darwinian mechanisms are responsible, it doesn't work nearly that fast. So the narrow window of time, the abruptness of the appearance of these organisms is really a puzzler. And in fact, of the phyla, there are between 13 and 16 of the phyla that arise within an even more narrow window of time, between 5 to 6 million years. So this is very mysterious from that standpoint. But so that raises this question, well, how would the evolutionary process have built these animals in such a narrow window of time, or even a much longer window of time? Well, it turns out that question has become very much more acute because of discoveries in the field of molecular biology, something completely unrelated. And we alluded to this last night. What are we talking about? We're talking about the discovery that at the foundation of life there is code, there is digital information. And if you want to build a new animal form, then you need to have new organs and tissues. And if you have organs and tissues, you need new types of cells, where each different type of cell, if you have a gut cell or a nerve cell or a skeletal cell, bone cell, you need specific types of proteins to service those types of cells. Like if you have a gut, then you need digestive enzymes. Those are specific proteins that are not present or activated in other types of cells. So you have this sort of a cascade of functional needs. You have an animal that has certain organs. Those organs require certain tissues. Those tissues require certain types of cells. Those certain types of cells require certain types of proteins. And where do we get proteins? I mentioned it last night, but we get proteins from the information in DNA. The DNA provides information for arranging the parts of the protein in a specific way. So proteins are long chain-like molecules, which many of you may remember from science class, and they're made of smaller subunits called amino acids. So I have this little visual aid here. I've got all my amino acid subunits. They link together, and if the amino acids are linked in the right sequence, they'll fold into a specific three-dimensional shape, and that three-dimensional shape will then allow the protein to perform a function by binding in a hand-in-glove way with other molecules in the cell. Okay, so there's a one-dimensional specificity that's required. You got to get the amino acids in the right arrangement, kind of like letters in an alphabet. If you want to spell a word, you can't just arrange the letters in any old way. They got to be arranged very precisely and specifically. And if that happens, then they'll fold into a specific three-dimensional shape that will allow the protein molecule to do a job. Okay, so every time you need a new cell type, it turns out you need dozens or hundreds or several new proteins to service that cell type. So if you're building new animals, you need lots of new cell types, lots of new proteins, and then lots of new instructions in DNA to build those proteins and cell types and tissues and organs and body plans. Is that making sense? Kind of a little bit of a science lesson here, okay? So and I remember my illustration from last night that the digital code in DNA is directing the construction of these protein machines and proteins. Now, next slide, please. I think we have a little illustration of it, some video of I get to move the slide, don't I? Sorry. Space cadet up here. Yeah, remember I talked last night about the sequence hypothesis of Francis Crick, the idea that the arrangement of the four subunits on DNA, they're represented with the letters A, T, G, and C. Those are called nucleotide bases. The arrangement of those nucleotide bases, which are functioning like alphabetic characters, that arrangement contains the instruction set for directing the construction of the proteins. And so that was the Crick sequence hypothesis. That's been confirmed. And now I have a little animation that shows how this is all done. So we'll now get a break from me talking. Well, exactly.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex. an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is often transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function.
So in the 19th century, in Darwin's time, they knew nothing about any of this. And one of his proponents, a man named Thomas Henry Huxley, who was said to be Darwin's bulldog, once said that the cell, the smallest unit of life, is a simple homogeneous globule of undifferentiated protoplasm. They thought of life as a simple goo enclosed by a cell membrane. So this is just one aspect of the complexity of life, but it's really relevant to our story here about the Cambrian explosion. Because if you want to build a new form of life, you've got to have information. If you think of it in the computer world, if you want to give your computer a new function, you have to provide it with new code. And the same thing is true in, I'm going to skip over a little bit of this technical stuff about different ideas about information. We can come back to that in discussion. But the big idea here for our discussion about the Cambrian period, if you want to build this Waptia guy from some simpler organism, maybe a sponge or a something in the Precambrian, you're going to need a lot of new information because you have to build the proteins to service the cell types, to service the tissues, to service the organs, and the organs and tissues have to be arranged somehow to make a body plan. So you got to have new information. Biological form requires biological information, okay? And so again, that raises the question we discussed last night a bit, which is where does that information come from? So just to give you an idea of the level of complexity, the jump, in the Precambrian there are these very primitive animals called sponges. They have between 6 and 10 cell types. A Cambrian fish has about 60 cell types. An arthropod, usually about 90 cell types. So you can actually cash that out in terms of number of nucleotide bases needed to build all the new proteins. It's a lot of new information. Where does it come from? Where would it come from? Now, the standard Darwinian explanation for the origin of new information during the process of biological evolution is that you have a gene, and then it is randomly changed. It accrues or collects random changes in its sequence of A's, C's, G's, and T's, the genetic letters, and those random mutations eventually produce a new gene that can produce a new protein that would be necessary to serve a cell type, etc. Okay? So you get random changes in an existing section of genetic text that cause a new sequence that's capable of building a new protein to arise. Now, there's a little bit of a problem with that, though, as we think about it. If you, in English, if we just take an English sentence like time and tide wait for no man, and we start randomly changing those letters, what's going to happen? Will you get a new English sentence? Well, if you change just a few of the letters, you might get still a recognizable word like time and tide bait no man or something. And maybe you've changed the meaning a little bit at first. But invariably, if you change a lot of letters, you're going to degrade the information that was present long before you'll generate anything new or functional or meaningful. Or how many people have some experience with coding, computer coding? Okay, so if you're a computer programmer and you have a section of of code that's performing a function within your hardware system, and you start randomly changing the zeros and ones, are you going to generate a new program or operating system, or are you going to destroy the function of the existing system? What's going to happen with a series of random changes? Well, programmers always laugh. It's obvious. Long before you get to a new program or operating system or something that's functional, you're going to degrade the existing function that you have by random changes. Now, this was recognized early on by some scientists from MIT. There was a famous conference in 1966 at the Wistar Institute in Philadelphia. It was called Mathematical Challenges to Neo-Darwinism. And it was convened by a group of MIT mathematicians, physicists, and computer scientists, and their biology colleagues. And it came about because there was a little picnic at one of the famous MIT scientists' house in 1965. His name was Victor Weisskopf. And the computer and maths and physics guys were talking to the biologists and said, wow, this is just amazing what you biologists are discovering about DNA and the gene expression system, how the DNA builds the proteins, which we just saw in the animation. That's fantastically interesting stuff. But we've got a question for you. If that's true, if that's what's really going on inside cells, how does your Darwinian theory work? How can a random mutation ever change that biological information into something new and useful? Aren't you going to destroy the information that's there already? And the biologist said, well, no, not necessarily. I mean, if there's enough time and enough opportunities, and the math-oriented scientists weren't having it. And one of them was Murray Eden, whom I met at a 40-year commemoration of that conference in 2007. And Eden said this, he said, no currently existing formal language can tolerate random symbol changes in the symbol sequences which express its sentences. Meaning is almost invariably destroyed. I know we've got a variety of backgrounds, but this is something that we can all understand regardless of science background, right? If you play Scrabble, okay, if you reach into the bag and try to pull out letters at random, you're very unlikely to get the triple word score, right? If you have a triple word score or some, functioning word, and you reach into the bag and you start replacing the letters, you're going to destroy the function of that sequence pretty quickly, okay? And this is what was worrying the math guys at MIT about what the biology guys were telling them. Okay, so they have a conference. Now, there's a reason that meaning is almost invariably destroyed. If you've got a sequence that's meaningful, let's just say a 12-letter English word. It turns out that with a 26-letter alphabet, there are 100 trillion possible ways of arranging those 26 letters into a 12-letter sequence. And of that 100 trillion, well, no, there's probably more than that. The point is, the ratio of the sequences that are meaningful that you can make with 12 letters to the sequences that give you gibberish is 1 over 100 trillion. Okay? So if you start changing things at random, are you more likely to find gibberish or meaning? You're overwhelmingly more likely to find the gibberish. All right, that's the problem. You're going to drop into a functionless abyss if you start randomly rearranging things. But what is the Darwinian mechanism? It's the idea that you randomly change the arrangement of the genetic letters and hope against hope to get a new sequence that is precise enough to generate a new protein that will be meaningful in a biological sense. And so the math guys at MIT said, that's implausible on its face. But let's do the math. Let's have a conference and do the math, okay? So they did. They had a conference. Here's an illustration of the kind of, another way of getting at this problem. Let's imagine that I have a 4-dial bike lock, okay? Oh yeah, this illustration actually, this is going to help us understand what came out of this conference in 66. So I've got a 4-dial bike lock, and I've got a beautiful racing bike locked up outside the church. And we're all in here, and there's a thief around the corner, and he realizes there's all these nice Christians in the church, and they're absorbed listening to this stimulating lecture, so I have an opportunity now to steal the bike. Is it more likely that the thief will succeed or fail in opening the lock at random? What's your guess? Okay, people are saying fail, but I'm going to tell you something now because I'm known to do this when I teach. It's a trick question, okay? Because there's something else you need to know. Now, you do know some things already, because if it's a 4-dial lock and it's got 10 digits on each, you know that there are 10,000 combinations. There's 10 times 10 times 10 times 10 possibilities. That's why a lock works, by the way. You're hiding the one thing you want among a bunch of gibberish sequences, and it's hard to find at random. Okay, so your intuition is right. Probably he's going to fail, but because there's a lot more ways to go wrong than there are to go right. Okay, just like the 12-letter English sequences. A lot more ways to go wrong than right. But we need to know something else. You need to know how long I'm going to drone on here. Because if I talk long enough and you're trapped here and they're in this audience listening to me, the thief's going to have more opportunities, right? He's going to have more opportunities to turn the dial. And I've done the math on this. You can do it yourself. But if the thief changes a possible combination, once every 10 seconds, in about 15 hours, the thief will have tried over 5,000 combinations. At which point, it becomes more likely that the thief will succeed than that the thief will fail. Okay. So in order to assess the plausibility of a random search finding a combination embedded in a large set of possibilities, you need to know how large is the set of possibilities, but you also need to know how many opportunities the thief has to try. Okay. So okay, if the thief has got this lock and 15 hours, because we lock the doors and keep everybody in here, it's more likely that the thief will succeed than it is that he'll fail. But what if we have this lock? What if we have a 10-dial lock? Okay, well, now I've done the math on this, and it turns out that if the thief changes the combination once every 10 seconds and does nothing but that for an entire 100-year lifespan, no potty breaks, no dinners, no anything, the thief will only get, even in that case, he'll only get to sample 3% of the possible combinations. In which case, it remains much more likely that he'll fail than it is that he'll succeed. Because it's a needle in a haystack problem at this point. He just doesn't have enough time to search enough of the haystack to have a reasonable chance of finding the needle. All right, in this case, the combination. Okay, so the question is, what is a gene, which is essentially like a lock, a long sequence of characters, is a functional gene necessary for building a functional protein more like the 4-dial lock case where the thief has 15 hours, or the 10-dial case where there's not enough time to search the spaces? Well, I have a colleague who did a PhD at Caltech named Douglas Axe. His PhD was in chemical engineering, but his thesis was on a molecular biological system, and he ended up becoming a protein scientist, and he did a 14-year postdoc at Cambridge University, and he asked a really important question. It's the question I just asked, only in more technical language. He said, how common or rare are the functional sequences among all the possible combinations of amino acids? So, let me back up. One more thing. If our 10-dial lock had lots and lots of combinations that would allow it to be opened, can you see that might change the odds? If it's 1 out of 10 billion, well, then it's going to be really hard to, but what if there's a billion of the 10 billion combinations that will open the lock? What if it's a really bad lock and it's not just one, but, almost, well, then in that case, you might have better odds. You might be able to by random means, find a series of functional combinations, okay? And that's what the biologists at Wistar were hoping for. They were saying, well, yeah, it looks improbable, but maybe there's lots of ways to build a functional gene or protein. Lots of arrangements will result in something functional. So Axe said, let's put that to the test. How many are there? Is it like the case of the one out of 10 billion? Or is it a case where maybe there's lots of combinations so that makes the odds better? And so he did a series of experiments at Cambridge over a 14-year period. They were called site-directed mutagenesis, technical thing, but basically he was sampling the different arrangements of A's, C's, G's, and T's on the DNA to see how many of them would allow you to get a functional, a folded structure that was capable of performing a biological function. And so he was asking for every one of these beautifully folded up structures that might perform a function in a cell, how many arrangements are there that are going to lie limp and not do anything for you? Molecular gibberish, okay? And the answer to that question was a really scary number. For every one that will fold a function-ready, it will have a function-ready structure, there's 10 to the 77 possible ways of arranging the amino acids that won't, that will be completely functionally inert within a cell. And that took into all the factors, the size of the space and the frequency of functional sequences within the space. Now, there was one other thing he needed to know, though, and that was, well, okay, that's a big haystack and a really small needle, but how many opportunities do you have to sample it? And so biologists actually have an estimate of that because they know how many organisms have lived on Earth and how many times the organisms, if you know the number of organisms, then you know the number of what are called replication events, how often the organisms would have a chance to copy themselves, and that's when mutations occur. So it turns out in the whole history of the planet, there have only been 10 to the 40th organisms. That's a lot of organisms. Most of those are bacterial species, which have no relevance to the Cambrian explosion, but that's a lot, but it's tiny in terms of 10 to the 77, the number 10 to the 77 possible combinations. So what that means, see if we can, don't want this to get too heady or technical, but are you guys following kind of the reasoning? Okay, it's really much like the biclock. It's just think of the biclock. So you've got a huge biclock, and you've got 10 to the 40th chances to search the different combinations, but basic math. Anyone here can do the division problem? 10 to the 40th divided by 10 to the 77? Thank you very much. 1 over 10 to the 37th power, okay? So that's like having a lock with 37 dials looking for one combination. That's what, mathematically, it's equivalent, that's what that number means. So that's, changing the metaphor slightly, very big haystack the size of California, very small needle the size of the pastor's office, okay? And you get to start blindly up somewhere in Fresno looking just anywhere in California for that one needle, that one combination that will open the lock, that one combination that will give you a functional protein. So this is much more like the second of my two. It's more like the 10-dial lock, only much, much worse. The bottom line is there's not nearly enough time from the beginning of life on planet Earth till now to have sampled the possible combinations via mutation to have a reasonable chance of finding one that will give you a functional protein. It's overwhelmingly more likely that that process of random mutation, sifted by natural selection, will fail in generating the biological information you need to build one protein than it is that it will succeed. Overwhelmingly more likely to fail than succeed. And in science, we don't like to bet on theories like that. If it's overwhelmingly more likely to fail than succeed, that means that the hypothesis that that's how it happened is overwhelmingly more likely to be false than true. Do you see that? And in science, we don't like to bet on hypotheses that are overwhelmingly more likely to be false than true. We want hypotheses that are more likely to be true than false. We want the best explanation, not a really bad explanation. And so that the random mutation sifted by natural selection explanation is a really bad explanation for the origin of biological information. That's the bottom line. But yet, what do we need to build a Cambrian animal? Or any new form of life? We need information. Okay, so that's the deal. So this is why you're finding that evolutionary biologists today are saying, we need a new theory of evolution. Mutation in natural selection is a real process. It can generate variations in coloration and finch beak size, but it doesn't have the creative power to generate the information that we need to build whole new forms of animal life. This is just very, very deep, profound, and fundamental. So Axe did his work from 1990 to 2004. He published his paper in the Journal of Molecular Biology. There has been other work since that time that's confirmed his results. There's an Israeli scientist who hates the idea of intelligent design, or rather hated it, I should say. He unfortunately passed away recently. His name is Dan Tawfik. Wonderful scientist, but he's no friend of intelligent design, but he's been quoted as saying that the probability of building a new protein fold, a structure that could perform a function by Darwinian means, he says it's essentially a miracle. We have no idea how this could have happened. And what he's shown is that if you have a section of genetic text that builds a functional protein, if you start randomly changing the genetic text, it takes about between 3 and 15 random changes to degrade what's called the thermodynamic stability of the protein. It simply won't fold anymore once you get past that 15 change threshold. So it's very much like the analogy I use to digital code. If you got a section of digital code, you start randomly changing the zeros and ones, you're more likely to degrade that information than you are ever to get to a new section of code that'll give you a new computer game or software program of some kind. And the same thing turns out to be true in life. You start randomly changing things, you degrade, you cause the protein to fall apart before you get to something new that will do a job in the cells. Okay, that's the hardest part of the lecture. How are we doing? Is this making sense? Okay. Just think of the bike locks. Okay, searching. Do you have enough time? You do not. You do not have enough time for reasonable success. Okay. Now, I'm just going to touch on this because it's so cool, and it's got a California angle on it. And then I'm going to get to why some of us are thinking that not only is neo-Darwinism a failed theory, but why there might be evidence in the living world of intelligent design. But something else that's been discovered about how you have to, if you want to build a Cambrian animal with all those new body parts and tissues and organs and cell types, you got to, well, it's a complicated process, it turns out. Any animal goes through, remember from biology when you have a fertilized egg, and so that's one cell, and then that egg divides and becomes 2 cells, and then those divide 4, 8, 16, 64, geometrically increases the number of cells. At a certain point, those cells start differentiating from each other. The different types of cells are needed to build an animal. You got to have bone cells, you have got muscle cells, you have nerve cells, you have to have cells for specialized tissues, for eyeballs and hair follicles, and each one of those cells has to be served, cell types has to be served by different kinds of proteins, which means different kinds of genes have to get turned on because you got the same genetic endowment in every one of those cells, but you only want some of the genes to be turned on to make some kind of proteins for a bone cell as opposed to a muscle cell. Okay, you need myosin in the muscles, and you need different proteins in different cell types. So that's a big question. Then how does the animal know, the developing embryo, know when to turn on certain parts of the genome and leave other parts shut off so that you get the right kinds of proteins being built in the right kinds of cells at the right time? It's a choreography problem. It's like a dance routine. And there's 2 amazing scientists at Caltech who worked on this throughout their whole career, and they ended up describing something called developmental gene regulatory networks. And the idea here is that there are specific genes, and signaling molecules that interact to form essentially an integrated circuit that controls and directs cell differentiation during animal development. So let me break it down a little. So maybe you're in you're a cell destined to become a muscle cell. So there's a gene that gets turned on because of a signaling molecule, and that turns on a certain gene, and that gene may in turn turn on another signaling molecule that'll shut off another part of the genome, but then that some other signaling molecule may turn on some part of the gene to express something else. And so there's this kind of interdependence of all of these. The genes turn on, they'll build proteins, but they also build what are called RNA regulators, regulatory molecules. So those molecules will turn on or turn off other parts of the gene at just the right time. And so this whole thing is carefully orchestrated so that you're turning on and turning off the right genes and and building the right proteins at the right time. And when Eric Davidson and his colleagues at Caltech mapped this out, this is what they got. They got something that looked like an integrated circuit. This is a diagram that explains the functional relationships between the genes and the gene products. That's pretty cool. It is an integrated circuit, except it's not mapping the flow of electricity, it's mapping a flow of information. It's a functional. Now, here's the problem, the other thing they discovered. There's a principle in engineering called the constraints principle. The more functionally integrated a system is, the more difficult it is to perturb, to mess with any part of the system without destroying the whole. Simple example, go out to your car, pop the hood, pull out a spark plug. No problem, you ought to be able to go home. You got 99% of the parts. But it's an integrated system, isn't it? That engine needs all of the parts to function. So you pull one out, you perturb one little tiny part of the system, cranking the key, nothing happens, okay? That's the constraints principle of engineering. It applies to this in spades. It's an integrated circuit. You start messing with any of the crucial elements of this, and there are many, and animal development shuts down. This whole thing is apparently set up to ensure that you go from that one cell to the full animal development. As soon as you start messing with any of the systems or subsystems or parts of one of these developmental gene regulatory networks, the animal, when we say development shuts down, that means it dies. That doesn't mean it lives on to participate in the evolutionary process. Evolution at that point terminates, okay? Now, here's the problem, and this was the one place I could have used a blackboard, but see if you can help me with you've got a developmental gene regulatory network that's represented by my hand. And it's going to build an animal. We'll put the little device up here, okay? Now, if you want to build a new animal over here that's different, that means you got to have a different developmental gene regulatory network. But what have we just learned about developmental gene regulatory networks? You can't change them without destroying animal development, without causing death. So how do you ever if you want to get from this animal to this animal, you got to get from this developmental gene regulatory network to a different developmental gene regulatory network. So if you can't change this one, how do you ever get from here to make this? Davidson says that this problem has not been solved by neo-Darwinism. He said neo-Darwinism is counterfactual to what we know about evolutionary or about embryological development. And it's not just neo-Darwinian theory. There are newer theories of evolution that are being proposed, and they don't touch this problem either. Nobody has a clue. But you have a circuit. Where have we seen circuits before? Who makes circuits? What kind of a cause could produce a circuit? Okay, we're getting warm now because we're getting to the idea of intelligent design. Now, he calls neo-Darwinism. This is Davidson, and he was at the conference in China in the year 2000, and he did everything he could to shun the participants there who were supportive of intelligent design. He hates religion. He passed away a couple years ago, but he was no friend of the idea of intelligent design or creationism or belief in God. But just as a scientist, he said that neo-Darwinism is a catastrophic error. I won't read the whole quote, it's in my book, but there are two other big problems with believing in the creative power of mutation and natural selection. I don't have time to go into them. But let's think about an entirely different approach. And this is kind of my story. I went to a conference not on the origin of animals, but on the origin of life when I was early in my scientific career. And there was a scientist there named Charles Thaxton who'd just written a book called The Mystery of Life's Origin, suggesting that the big problem with the origin of life was the origin of all that information you need to build a cell or to build an animal. And he suggested that maybe we're kind of barking up the wrong tree, that when we look at information, we know from our experience that information is a mind product. It's not a product of material processes. It's code in a software program comes from a programmer. So maybe we ought to be thinking about opening our minds to the possibility that when we're looking at these complex informational systems in cells and animals, we're really looking at evidence of what he called an intelligent cause. Now, I got fascinated with that idea. I met him after the conference. He started mentoring me. I'd go to his office late at night. We'd talk for hours. I was newly married. My poor wife always wondered, where is he? What's happened? It was before cell phones. Anyway, a year later, I was off to grad school in Cambridge, and I had decided I wanted to work on this question of the origin of the first life. And I had kind of a burning question in my mind, and that was, could the design hypothesis, could this idea of an intelligent cause being part of the explanation for the origin of life, could that be formulated as a rigorous scientific argument? And this was my mentor, Charles Thaxton. He's still alive. He's just published his autobiography. He is a cancer survivor. He had a leg amputated. His book is whimsically titled A Leg to Stand On. He's a wonderful man. In any case, I got to Cambridge wondering, could you develop the case for an intelligent cause, or what we now call intelligent design? Could you develop that in a rigorously scientific way? Could you make a scientific case for intelligent design? Now, oddly, the person that helped me think that through more than anyone else was Charles Darwin, another Charles. Because he pointed out that if you're trying to reconstruct what happened in the ancient past, if you want to know what caused an event to happen in the ancient past, you have to use a different method of reasoning. You're not in the laboratory. You can't make trilobites arise in the laboratory under controlled laboratory conditions. You can't even build a cell in the lab under controlled laboratory conditions. So we don't know how to do that. So we've got to use a different method of science. We've got to be smart about this. We've got to reason like forensic scientists or detectives. We have to look at the clues that are around us, and then we have to think about what might have caused those clues based on what we know about the cause and effect structure of the world. And so I'm going to skip over this. Another Charles comes into this story too. This was Darwin's mentor. He was the great geologist, Charles Lyell. And I was one day reading the front piece of his great work of geology, The Principles of Geology. This was the title page. There's a long, boring Victorian subtitle. Being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation. I suddenly woke up. Bing, light went on. If we want to explain an event in the remote past, what caused that event to happen, we want to know what kind of causes now in operation in the present are capable of producing that event. Let me give you an example. I went to college in Eastern Washington at Little Whitworth University. In Eastern Washington, there's this stuff called Palouse. It's the wind-blown sediment where they grow all the wheat. And if you weren't around on May 18th, 1980 to know what happened, you might be able to figure it out from some clues that have been left behind. There are little patches of white powdery stuff, all still sprinkled across the Palouse country. So if you're using the Lyell or Darwin method, you go and you look at the white powdery stuff and you're going to say, okay, what is a cause now in operation that could produce white powdery stuff? And you then use a method known as multiple competing hypotheses. You say, well, maybe it was an earthquake. Maybe it was a flood. Maybe it was a rainstorm. Maybe it was a volcanic eruption. Of those 4 possible causes, which provides the best explanation for the white powdery stuff that's still on the ground in eastern Washington? Well, earthquakes don't produce white powdery stuff. Rainstorms don't produce white powdery stuff. Windstorms don't produce white powdery stuff, but volcanic eruptions do. That's a cause now in operation that produces the effect we're trying to explain. So the best explanation becomes the volcanic eruption, and then we can infer from that white powdery stuff to an event that happened before it. That's the historical method of reasoning that Darwin developed, building on Lyell's work. So I got to thinking about that. Could that be used to investigate the problem of the origin of life or the origin of biological information? And so I asked myself another question. What is the cause now in operation that produces digital code? As I was formulating that question, I was reading something else. I talked about this last night. I have a little book on the application of information theoretic concepts to molecular biology. And the scientist who wrote this, Henry Quasler, who was a pioneer in this field, said, in an offhand, I'm reading this, is a major gestalt aha moment for me. Page 16, I can see it on the page. The creation of new information is habitually associated with conscious activity. What's he saying? To use Lyell's words, he's saying that information, that conscious activity is a cause now in operation for producing digital code. So I thought, oh, that means you could use the Darwinian and Lyellian method to infer intelligent design. Because our uniform and repeated experience of cause and effect shows that the only known cause of the generation of digital information is a mind, is intelligence, is conscious activity. I used this quote last night, DNA is like a computer program, but far more advanced than any we've ever created. Where do computer programs come from? From programmers. In other words, I'm asking the question, is Henry Quasler right? Is conscious activity the only known cause of digital code? Well, think about it. Computer programs come from programmers. Paragraphs in books come from writers. Hieroglyphic inscriptions come from scribes. We know of no exception to this rule. In fact, the people that are looking for extraterrestrial intelligence in space, they have those big satellite dishes and they're taking in all the signals from space. What are they looking for that would tip them off to ET? They're looking for information embedded in a radio signal. In the SETI, in the Contact movie that was out years ago, when they found the prime number sequence embedded in this, that's an information sequence. That's not just a random thing. That's something that only a mind would know about. So information in our experience, thank you very much, is an indicator of prior intelligent activity. That's the bottom line. Now, by the way, so are circuits. Do we know of any other cause of the origin of circuits other than engineers, other than minds? No. So we have indicators in living systems that I argue are pointing, there's more that we skipped over, that point to intelligent design. Now remember, to build a Cambrian animal, what most fundamentally do you need? New organs, new tissues, new cell types, new proteins, but most fundamentally, you need the information to build all those things. So when we look at an event like the Cambrian explosion, where you see a huge burst of biological form, and we know that that form could only arise with an equally huge burst of information, we are looking at evidence, I argue, for the activity of a designing mind in the history of life. And notice that in coming to that conclusion, we're using the same method of reasoning, the same scientific method of reasoning that Darwin himself used in The Origin of Species. The technical name for it is inference to the best explanation, or it's sometimes called the method of multiple competing hypotheses, where the best explanation is the one that offers a cause which is known to produce the effect in question when no other such cause is known. And that's precisely what we have when we're talking about information, the information needed to build life. So that's my case for intelligent design. Now, just in closing, you might want to know, what my critics have said. I had a wonderful supervisor in Cambridge who used to say, "Beware the sound of one hand clapping." He said that was his way of saying if there's an argument on one side, there's bound to be an argument on the other. And what follows from that is that if you want to test the strength of an idea, you want to see what the critics of the idea are saying, okay? So let's just do that so you guys, this will set up our Q&A discussion time. This is a man I respect greatly. I cited many of his works of paleontology in my book, Darwin's Doubt. His name is Charles Marshall. He's a Kiwi, he's from New Zealand, but he's a professor at Berkeley. And he reviewed my book, Darwin's Doubt, in Science. And he was complimentary in some ways. He said it was very nicely well written. He had some other nice things to say, but he was ultimately critical. And this was his criticism. He says that Meyer's case depends upon the claim that the origin of new animal body plans requires vast amounts of novel genetic information. In fact, that means Meyer's not right about that. In fact, our present understanding of morphogenesis, of body plan building, indicates that the new phyla were not made by new genes, but largely emerged through the rewiring of the gene regulatory networks of already existing genes. Can anyone help me break that down? What do you think of that critique? Well, okay, the rewiring, if you're an engineer, gets a little suspicious. What did we just learn about developmental gene regulatory networks? You can't change them. You can't rewire them without breaking them. Okay, that's problem one. Number two is the gene regulatory networks are a system of already existing genetic information and their RNA and protein products. So I'm saying you need information to build these new Cambrian animals. He's saying, no, you don't. You just need these developmental gene regulatory networks and you got to rewire them a little bit. But the gene regulatory networks have a huge genetic endowment. They already have information in them and he's not explained the origin of that information. So he's begged the question as to the origin of the information in the gene regulatory network. And then he says, he also says that but we're largely emerged through the rewiring of the gene regulatory networks of already existing genes, okay? So there's also there's some other, and I break this down in my response, he's begging the question of the origin of yet other genetic information. You need information for building each of the parts of the Cambrian animals, and then you need information in the gene regulatory networks to arrange and turn on the cell types to build those parts at the right times. So so I responded to his critique in print, and I made 3 points. He's asserting something about gene regulatory networks which is contrary to what we know experimentally. You can't rewire them. To rewire them would be to rearrange modular elements, which would require information, and even the metaphor implies intelligent design. And then he's presupposing the existence, he's presupposing the existence of two sources of unexplained genetic information, the origin of which he is not explaining. Okay, and so we ended up having a debate on a British radio for 90 minutes. And I pointed this out, that you could try to push the origin of the Cambrian animals back into the Cambrian with some hypothetical organism that already had all this information, but then you still hadn't solved the problem, which is the origin of the information in the first place. And in his Kiwi accent, he said, well, fair comment. So I thought that was good. Anyway, he's a great guy. He's a really smart scientist. I think he's just up against it like all the Darwinian biologists are, the neo-Darwinian biologists and others. I think Marshall's already entertaining new theories of evolution. But the problem is, the fundamental problem of the origin of information and form has not been solved by any leading evolutionary mechanism or theory. And that's where we are. Okay, let's stop there. And thank you for your attention. This was obviously more detailed than last night, more scientific. Yeah.